Hi, my name is Dr. Jamie Caroon. I am the medical director at the Center for Medical Cannabis Education in San Diego, California. In this video, we are going to look at the first study to demonstrate that CBD can increase blood flow in the hippocampus, a part of the brain that is associated with memory. This study is entitled The Effects of Acute Cannabidiol on Cerebral Blood Flow and its Relationship to Memory, an Arterial Spin Labeling Magnetic Resonance Imaging Study. It was published in the Journal of Psychopharmacology in August of 2020. Before I talk about this study, I'd just like to point something out that we see far too frequently online. So the title of this article on CBDinsider.com states that CBD can improve memory function, according to a study. And then here are the results as stated by the investigators. There were no differences in memory task performance. So it's really important to remember that there is so much hype surrounding CBD right now, and only time will tell how much of it is unfounded. And until then, it's wise to be very critical of what you hear and what you read, and to consider the source. This happens a lot, including in mainstream media publications. So with that public service announcement, let's get into this study. It was a randomized, crossover, double-blind, placebo-controlled trial. The study population was comprised of 15 healthy adults. The intervention was 600 milligrams of CBD administered orally, and the primary outcome was cerebral blood flow, abbreviated CBF, in regions of the brain that are thought to be involved in memory, specifically the medial temporal lobe and the prefrontal cortex. And this cerebral blood flow was assessed using a functional MRI with arterial spin labeling, which is a very precise method for detecting changes in blood flow. Secondary outcomes were memory-related, specifically memory performance in both episodic and working memory tasks. It was implemented by investigators at the Department of Mental Health Neuroscience at the University College London. By way of background, preclinical models have suggested that CBD may improve working and episodic memory, including in the presence of memory impairment. That's both THC-induced memory impairment and pathological memory impairment, but these are preclinical models. The precise mechanism underlying these effects are unclear. There is evidence that CBD can alter cerebral blood flow, but no study has investigated the association between regional cerebral blood flow and memory task performance following acute administration of CBD. As I mentioned previously, this was a randomized, crossover, double-blind, placebo-controlled trial with 15 participants. They were given CBD and placebo by capsule with 1 week in between. The brain imaging took place 3 hours after ingestion. Memory tasks were performed in succession from between 4.5 to 5.5 hours after ingestion of the CBD. And one interesting point regarding the eligibility criteria: participants could not have used cannabis or CBD at all in the past. So these participants were totally naive to CBD. Participants served as their own controls, which is how it works with a crossover study. The study population was mostly comprised of females in their mid-20s. Average age was 24. The study drug was provided by STI Pharmaceuticals, a UK-based pharmaceutical company. It was 99.9% CBD. To be clear, this was not a full-spectrum cannabis extract. It was isolated CBD. Participants took 2 capsules and entered the functional MRI tube 3 hours later. A quick overview of the data analyses, and finally getting to the outcomes. Okay, let's get into the results. This is most of Table 2 from the study. Here are the 6 regions of interest on the left-hand column. The next column to the right shows the change in perfusion. The third column shows the percent change in perfusion. The fourth column shows the Cohen's d, or the effect size. And then the final column shows the corrected p-value. So these are the p-values corrected using Bonferroni or home Bonferroni. As you can see, CBD caused a significant increase in cerebral blood flow to the hippocampus with a medium to large effect size according to the Cohen's d value of 0.75. CBD did not cause significant differences in cerebral blood flow in any of the other regions. There was a statistically significant increase in the orbitofrontal cortex, but that did not survive the home Bonferroni correction. So once it was corrected for multiple comparisons, it was no longer statistically significant. Now, these post hoc corrections are important because they reduce the type 1 error rate, or the false positive error rate. And so as a result, the threshold for determining statistical significance becomes more difficult the more corrections you perform, because the possibility or probability of a false positive error increases with each additional comparison. The secondary outcomes were memory related. Here we see differences between CBD in blue and placebo in red across the various memory tasks. So just a quick review of the heights of these bars indicates that they are basically the same height, which suggests that there is very little difference between CBD and placebo. A strong correlation was found, however, between one aspect of one memory task and one region of the brain. But this was just a correlation, so it's not clear that the increase in memory performance was actually caused by CBD. As you might expect, the plasma levels of CBD were significantly higher when participants took CBD. The investigators did not mention the bioavailability in percent terms, but historically it's quite low for isolated CBD, around 10% or sometimes even less. Adverse events were not reported in the paper. With regard to limitations, we have to acknowledge that this was one pretty high dose of CBD given to healthy adults with a wide range of responses. And this is really important because in all of these studies, you see so much biological variability between the participants. And that could be anything from bioavailability to cerebral blood flow. It was only powered to detect a large effect size, so Cohen's d of 0.8 or higher. So it's possible that important differences were not detected because the study was not powered to detect an effect size that was less than 0.8. In conclusion, this is the first study to find that acute CBD administration orally increases cerebral blood flow in the hippocampus of humans, although it didn't seem to help with memory. So this is acute administration in naive participants. It doesn't necessarily mean that if you take CBD on an ongoing or chronic basis, this effect will be sustained. And the findings agree with a meta-analysis of a mouse model of stroke, but they disagree with 2 previous human studies which used a different imaging technology. They used SPECT instead of MRI, which is less sensitive, and it was shown to decrease cerebral blood flow in those studies. This study has a greater sample size and a greater power than those previous studies did. And really what it comes down to is that we need more randomized controlled trials to better understand the impact of CBD on cerebral blood flow. We need trials with larger sample sizes and a bunch of different populations, healthy adults, adults with anxiety, children, etc. And we need a bunch of different dosages of CBD in these trials. And then we also need to understand if CBD in a full spectrum extract has the same effects as isolated CBD. And so it's difficult to know whether we can extrapolate these results to products that are available over the counter. And finally, it's important to remember that changes in cerebral blood flow do not necessarily translate into meaningful clinical outcomes, which is what really matters. With that, I'd like to thank you for your time and your attention. If you have any questions or feedback, please email me at jamie@karoon.com. You can find more videos like this at drjournalclub.com.